Hello, welcome to Biology, the scientific study of life. DNA replication, a closer look. The bacterium E. coli has a single chromosome of about 4.6 million nucleotide pairs in a favorable environment. An E. coli cell can copy all this DNA and divide to form two genetically identical daughter cells in less than an hour. Each of your cells has 46 DNA molecules in its nucleus, one long double helical molecule per chromosome in all. That represents about 6 billion base pairs, or over 1,000 times more DNA than is found in a bacterial cell. If we were to print the one-letter symbols for these bases A, G, C, and T, the size of the letters you are now reading, the 6 billion bases of information in a diploid human cell would fill about 1,200 books as thick as this text. Notice that. Yet it takes a cell just a few hours to copy all this DNA. This replication of an enormous amount of genetic information is achieved with very few errors, only about 1 per 10 billion nucleotides. The copying of DNA is remarkable in its speed and accuracy. More than a dozen enzymes and other proteins participate in DNA replication. Much more is known about how this replication machine works in bacteria than in eukaryotes. Back to inquiry: does DNA replication follow the conservative, semi-conservative, or dispersive model? Notice the experiment. Matthew Michelson and Franklin Stahl cultured E. coli bacteria for several generations on a medium containing nucleotide precursors labeled with a heavy isotopes of nitrogen, N to the power 15. The bacteria incorporated the heavy nitrogen into their DNA. The scientists then transferred the bacteria to a medium with only nitrogen to the power 40. The later more common isotopes, notice that, of nitrogen. Any new DNA that the bacteria synthesized would be lighter than the parental DNA made in the N to the power 15 medium. Michelson and Stahl could distinguish DNA of different densities by centrifuging DNA extracted from the bacteria. Notice that a bacteria cultured in medium containing into the power 15, and this one bacteria transferred to the medium containing into the power 14. Results: DNA sample centrifuge after 20 minutes after first replication, and this one DNA sample centrifuge after 40 minutes after second replication. Less dense and more dense. Notice that the bands in these two centrifuge tubes represents the results of centrifuge 2 DNA samples from the flax in steps 2 on sample taken after 20 minutes and one after 40 minutes, as is shown here. Conclusion: Michelson and Stahl concluded that DNA replication follows the semi-conservative model by comparing their results to the results predicted by each of the three models, and in the figure this one. The first replication in the N to the power 14 medium produced a band of hybrid N to the power 15 minus N to the power 14 nitrogen, which means DNA. This result eliminated the conservative model. A second replication produced both light and hybrid DNA, a result that eliminated the dispersive, notice that, model and supported the semi-conservative model. Conservative model, first replication and second replication, notice, and this one is semi-conservative model, and the last one dispersive model, notice the bon. Okay, thanks. Bon. Let's continue getting started origins of replication. The replication of a DNA molecule begins at special sites called origins of replication. The bacterial chromosome, which is circular, has a single origin, a stretch of DNA having a specific sequence of nucleotides, proteins that initiate DNA replication, recognize this sequence and attach to the DNA, separating the two strands and opening up a replication bubble. Replication of DNA then proceeds in both directions until the entire molecule is copied. Notice that. Also, in contrast to a bacteria chromosome, a eukaryotic chromosome may have hundreds or even thousands of replication origins. Multiple replication bubbles form and eventually fuse, thus speeding up the copying of the very long DNA molecules. Notice that. Oh. As in bacteria, eukaryotic DNA replication proceeds in both directions from each origin. At each end of the replication bubble is a replication fork, a Y-shaped region where the new strands of DNA are elongating. Elongating a new DNA strand. Elongation of new DNA at a replication fork is catalyzed by enzymes called DNA polymerase. As individual nucleotides align with complementary nucleotides along a template strands of DNA, DNA polymerase adds them on one by one to the growing end of the new DNA strand. The rate of elongating is about 500 nucleotides per second in bacteria and 50% or 50 per second in human cells. In E. coli, two different DNA polymers are evolved in replication: DNA polymerase third and DNA polymerase 1. The situation in eukaryotes is more complicated, with at least second different DNA polymers discovered so far. However, the general principle are the same. Each nucleotide that is added to a growing DNA strand in actually a nucleoside, a triphosphate, which is a nucleoside, sugar, and a base. With three phosphate groups, we have already encountered such a molecule, ATP, adenosine triphosphate. The only difference between the ATP of energy metabolism and the nucleoside triphosphate that supplies adenine to DNA is the sugar component, which is deoxyribose in the building block of DNA. But ribose in ATP, like ATP, the triphosphate monomers used for DNA synthesis are chemically reactive, partly because their triphosphate tails have an unstable cluster of negative charge. As each monomer have an unstable cluster of negative charge, as each monomer joins to the growing end of a DNA strand, it loses two phosphate groups as a molecule of pyrophosphate PP. Subsequent hydrolysis of the pyrophosphate of two molecules of inorganic. Notice that. Phosphate P is the exorganic reaction that drives the polymerization reaction. Notice that the new strand and the template strand. Incorporation of a nucleotide into a DNA strand. DNA polymers catalyzes the addition of nucleoside triphosphate to the three ends of a growing DNA strand. Notice that. Three and 5 and the opposite. Okay, thanks for